Hello and welcome to another episode of Momento Malum today. The high bush blueberry is a species of perennial shrub, native to North America. This fruit species is currently cultivated across North America, Oceania, and Europe. It is the primary blueberry species cultivated for its fruit. The high bush blueberry has many lineages in the wild that come in many different forms such as diploid, tetraploid, and hexaploid. The high bush blueberry was domesticated in the 20th century. The first breeding program for the predominant type, known as the northern high bush, was initiated by Dr. F. Coville of the USDA. The primary founder populations were selections from the wild, specifically the cultivars Brooks, Soy, and Rubel. These are found in the pedigree of 23.5, 12.5, and 28.6% of the blueberry cultivars, respectively. From 1908 to 1994, all the cultivars produced were descendants from these three initial wild samples, through crossbreeding with other wild specimens or selections. From 1994 onward, other species were incorporated into the general blueberry gene pool. In the north, the low bush blueberry would be incorporated into the general blueberry gene pool through the cultivars Russell and North Sedgwick. In the south, the scrub blueberry, black blueberry, and rabbit-eyed blueberry would be incorporated into the general blueberry gene pool to create the southern high bush blueberry gene pool. In order to figure out the genetic diversity of the cultivated blueberry gene pool, the researchers who are referenced within this paper utilize the SSR marker technology to analyze 69 genetic lines of blueberries. These include 12 wild specimens, bred prior to 1950, 2 founder cultivars, 15 modern northern cultivars, 14 modern southern cultivars, 1 pentaploid cultivar, 1 half-high cultivar, and 4 hybrids between southern and northern cultivars. The results found that there are more unique genetic regions in the wild populations of blueberry in comparison to the cultivated blueberry. Blueberry cultivars and wild specimens split into 2 main groups, with each group split into different clusters. The wild lines were clustered in accordance to their geographical location, where the specific line was discovered. Of the northern high bush blueberries, all fall into one cluster and subdivide into 3 subclusters. Subcluster 1 consists of descendants of the cultivar Stanley and Stanley itself. Subcluster 2 consists of the cultivar Cabot and those of its line. Subcluster 3 consists of cultivars that are derived from subcluster 1. The cultivars of the southern high bush blueberries all fall into one cluster, but it's not fully known how they divide into subclusters. However, it is known that the cultivars Magnolia, Misty, and Blacksea all group together into one single subcluster through their shared pedigree through Avon Blue. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below. There is very little difference in terms of genetic diversity between the northern and southern lineages. The most genetically unique lines in the southern and northern pedigree groups were found in the oldest cultivars such as Cabot, Olympia, Blue Jay, Florida Blue, Taylor, and Misty. There are some exceptions, of course. The half-high cultivars have greater pedigree in the low bush blueberry species, and as such have greater levels of genetic differentiation from the rest of the high bush blueberries. Southern lineages show less genetic diversity and genetic uniqueness due to being frequently backcrossed into the primordial high bush cultivars. Northern lines, on the other hand, show a greater intermixing between clusters, including mixing between southern and northern lines. We will conclude with the pedigree of the listed cultivars utilized in the reference paper, and as per usual, the paper can be found below in the description below. Thank you for watching.